All right, let's dive right into the deep end, to the very edge of what we even consider life. We're going to explore this really bizarre group of, well, entities that live in that gray area between living and non-living. I'm talking about the ultimate parasites, viruses, viroids, and prions.
And this really gets us to the central question, one that's fascinated biologists for, a century. Are these things alive? I mean, they can replicate, they can evolve, but stick one outside of a host cell and it's basically as inert as a grain of salt. So what gives?
Okay, so the one thing that unites all of them is that they are acellular. That means they're not made of cells. This just shatters one of the most fundamental rules of biology, right? Cell theory, the idea that the cell is the basic unit of all life. Well, these guys, they play by a completely different set of rules.
Think about it like this. Cellular life, like you, me, or a bacterium, is kind of self-sufficient. We have our own metabolism, we reproduce by dividing, but these acellular agents, they're totally inert outside a host. They don't grow, they don't divide. Instead, they hijack a host cell and basically turn it into a factory that's forced to assemble new copies of them. It is pure molecular parasitism at its most brutal.
So, let's kick off our journey with the one you probably know best, the most complex of the three, the virus. These are the true architects of infection.
So what is a virus really? The textbook definition is an obligate intracellular parasite. Yeah, it's a mouthful, but it just means it absolutely has to be inside a living cell to do anything. That's it. It's basically just a little piece of genetic material, could be DNA, could be RNA, wrapped up in a protein shell called a capsid. And it has one job, and one job only: get its genome into a host cell. And wow, have they evolved some incredible structures to pull that off. You've got simple helical rods, you've got these cool icosahedral shapes, kind of like a 20-sided die, and then you have the complex viruses, like bacteriophages. I mean, these things look like they flew right out of a sci-fi movie. They literally function like a little molecular syringe, latching onto a bacterium and injecting their DNA straight inside. It's amazing.
Okay, so how do they actually replicate once they're inside? Well, there's this brilliant system called the Baltimore classification, and it organizes all known viruses based on a single elegant question: how do you make your messenger RNA? Because once you have mRNA, you can make proteins. So whether you're a DNA virus like herpes or a retrovirus like HIV, you've got your own unique strategy to hijack the host's protein-making factory. Now, for those bacteriophages we talked about, the takeover can go down two main paths. First, you have the lytic cycle. This is the full frontal assault. It's get in, replicate like crazy, and burst the cell open, releasing hundreds of new viruses. Boom. Then there's the lysogenic cycle. This is the stealth mission, the long game. The viral DNA just slips right into the host chromosome, lying low, getting copied every single time the cell divides, just waiting for the perfect moment to strike.
Okay, so now we're going to start stripping away the complexity. Let's take that virus and get rid of its protective protein coat. What are you left with? Well, that brings us to the viroid. I mean, really think about that. What if you just had a piece of infectious genetic material, all on its own? Could that actually cause a disease? For the longest time, the scientific consensus was no way. But as it turns out, nature had a pretty big surprise up its sleeve.
So, back in the late 60s, scientists were totally stumped by this potato disease. They knew something was causing it, but it was smaller than any virus they'd ever seen. It took until 1971 for a researcher named Theodore Diner to finally figure it out. He realized he was looking at something completely new, just a tiny, naked, infectious loop of RNA, and he gave it a name, the viroid.
And the properties of these viroids are just bizarre. They are the smallest known pathogens, period. Just a short, circular strand of RNA, no protein code at all. But here's the craziest part. Their genome doesn't code for a single protein. So you have to wonder, how on earth does it replicate? How does it cause disease?
It's an absolutely masterful deception. First, the viroid tricks a host enzyme into making copies of itself. But the real genius is in how it causes the disease. You see, the plant's immune system sees this foreign RNA and chops it up into little pieces to target it for destruction. But some of those little pieces are a perfect match for the plant's own essential genes. The viroid literally tricks the plant into silencing itself. It turns the plant's own defenses against it. Incredible.
Okay, so we've stripped away the protein coat. Now for the final, most radical step in this journey of simplification. What if we get rid of the genetic material, the DNA, the RNA, all together. Can you still have an infectious agent? This brings us to the most mysterious and, frankly, terrifying agent of all, the prion.
When a scientist named Stanley Prusiner proposed what he called the protein-only hypothesis, it was practically heresy. It flew in the face of the central dogma of biology, the idea that an infection could be spread by a protein, just a protein, with zero genetic instructions. It was so revolutionary that it eventually won him the Nobel Prize.
So how in the world does this work? It's basically a tale of two shapes. We all have this normal, harmless protein in our brains called PRPC, but there's an abnormal, infectious version called PRPSC, and here's the key. They're made of the exact same stuff, the same amino acids, but they're folded into a different 3D shape. The normal one is fine, harmless. But the misfolded one? It's incredibly stable. It loves to clump together, and it's highly toxic to brain cells.
And this is the really terrifying part. When one of these bad, infectious prions touches a normal one, it acts like a template and forces the normal one to misfold into the bad shape. It's like a zombie protein converting a healthy one, and this sets off a chain reaction, an exponential cascade of misfolding. These newly converted zombie proteins then clump together into these massive, stable plaques that just wreck brain tissue, leaving it riddled with sponge-like holes. And this one single terrifying mechanism is the cause of a whole spectrum of fatal diseases. You've got scrapie in sheep and goats, there's chronic wasting disease in deer and elk, and of course, the one everyone's heard of, bovine spongiform encephalopathy, or mad cow disease. In humans, it's responsible for things like Creutzfeldt-Jakob disease and Kuru, a disease famously linked to ritual cannibalism in Papua New Guinea.
Okay, so there you have it. We've met our three agents: a genetic parasite wearing a protein coat, a naked RNA parasite, and a shape-shifting protein parasite. Let's pull them all back together for one final showdown.
And here it is, side by side. You can see it really clearly. Viruses are made of nucleic acid plus protein, and they can infect pretty much all life. Then you have viroids, which are just RNA, and they stick to infecting plants. And finally, prions, which are only protein, and they seem to target mammals. I mean, look at that. Three totally different compositions, three unique and frankly brilliant strategies for survival, all operating right on that strange blurry boundary between chemistry and life.
So, where does this leave us? What does it all mean? Well, for one, these agents prove that replication and evolution can happen without cells, which is already a huge deal. But the prion shows us that heritable information, the kind of stuff passed from one generation to the next, can be encoded not in a gene, but in a physical shape. And that leaves us with a much bigger question than just, are they alive? It forces us to ask, if a protein's shape can be infectious, what other fundamental things about the nature of life itself are we still completely missing?